Okay, thank you. Yes, my name is Adam Dixon, and I'm from the University of Virginia. And today I'm presenting primarily Johnny Chen's work. He's done about 99% of this, but I'm giving the presentation today. Oops. Okay. And the title of the talk is Acoustically Active Red Blood Cell Carriers for Ultrasound-Triggered Drug Delivery with Photoacoustic Tracking. So we saw a very nice example in earlier talks of ultrasound-mediated drug delivery. But if we consider some conventional agents for ultrasound-mediated drug delivery, we can think of perfluorocarbon droplets and microbubbles. And unless you employ a strategy like we've seen earlier today, these agents have a limited drug carrying capacity, primarily due to the small volume of their shell. However, for about 30 years, red blood cell carriers have been studied to deliver drugs. Essentially, the drug is encapsulated inside the red blood cell's membrane. These offer very large drug carrying capacities, and they're inherently biocompatible. And however, these drug carriers typically release their drug via diffusion and slow release. So they're not activatable in the sense that these agents are. So on the left, we have an activatable agent with a small drug carrying capacity. And on the right, we have a non-activatable agent with a very large drug carrying capacity. So the idea for this work was to actually combine these two agents. So what we've done is we have a perfluorocarbon droplet with some drug encapsulated into a red blood cell carrier. And so we're calling these acoustically activatable red blood cell carriers. And so that we can achieve a localized delivery from these red blood cells and then potentially reduce the drug dose and potentially reduce side effects. And so the way that we synthesize these agents is we isolate red blood cells, and then we expose the red blood cells to a hypotonic solutions. And so in doing so, we actually induce pores in the red blood cell membrane due to the difference in osmotic pressure. And so hemoglobin will leave the red blood cell, and this is the opportunity to load a drug, or in the case of this study, we're using calcine as a model drug loaded into the red blood cell. And then we can reseal the red blood cell membrane by exposing it to an isotonic saline solution. And then in this work, we're attaching the perfluorocarbon nanodroplets via an electrostatic interaction. The perfluorocarbon droplets have a cationic shell. And so here's an example, some fluorescence images of the agents that we've created. In the bright field, the large circles here are the red blood cells. And you can see these smaller circles here are the droplets. We've incorporated the fluorescent molecule di into the droplet shell, so you can see individual droplets. We've loaded the calcine into the red blood cells, so this is the calcine channel. And then when we do the overlay, you can see that there are droplets in contact with the red blood cells. And so, as I stated, the overall goal here is to intravenously inject these red blood cell carriers and then insonify them to convert the droplets and then release the drug. Or again, in our case, we're using calcine. And so I have a video here of this effect. So again, this is a dual channel fluorescent image here. So the green is the calcine encapsulated in the red blood cell. And then the red, you can faintly see it here as a droplet, and there's another droplet over here. And as I play the video, oops, let me go back. As I play the video, we will apply a 10-cycle, 10 megahertz ultrasound pulse at, again, a very high peak negative pressure, so 11 megapascals. So let's, where's my mouse? Okay, I had this trouble earlier, but this seemed to fix it. So let me try this again. Sorry for the delay. So the ultrasound comes in, and you can see actually that we ruptured the red blood cell here, and we've released the calcine. So it looks like the droplet's remaining. However, the channel for the fluorescence of the droplet was taken before the experiment, and we just have it overlaid on this image. So the droplet is also converted after the exposure of the ultrasound. So this confirms that we can have activatable release. And then so as a second study, we decided to try to quantify the release here. So we've place the red blood cell carriers in a tube, and we're doing a 10-minute insonification at similar ultrasound parameters. And then we measure the fluorescence in the supernatant that's released from the red blood cells, and we can compute a percent release. And so when we do this, we have percent release on the y-axis here. When we don't apply ultrasound, we do see some release of the calcine. This is because the calcine slowly leaks out of the red blood cell membrane. In the resealing process, we don't completely close every single pore. However, when we introduce ultrasound, we have perfluoropentane droplets and perfluorobutane droplets, and we see between 30 to 50% release of the fluorophore. And as I mentioned earlier, we actually have the capacity to have a very large payload in these red blood cells. And so we've experimented with putting magnetic nanoparticles into the red blood cell core, so loading that along with the calcine. And you can see we have a tube here where the red blood cells are homogeneously distributed. Then we introduce a very strong rare earth magnet. And then after about 10 minutes, the red blood cells are attracted to the magnet. So this can be yet another layer of potentially aggregating the red blood cells at a therapeutic site. And so, again, we have a large payload, so we can put a lot of things in these. If we want to do real-time image guidance or image tracking of these agents, we basically cannot distinguish between the ultrasound signal from a native red blood cell or these red blood cell carriers. So instead, we've elected to load a dye into the inside of the red blood cell carrier and then use photoacoustic imaging to track these using photoacoustics. And so one interesting thing here is we should be able to track these without actually destroying the destroying this acoustically activatable RBC, because the droplets will not convert when we shine the laser light. So it's kind of like a non-invasive tracking mechanism. And so we've done this with indocyanine green. So we've loaded ICG into these red blood cells. And then we use a linear array photoacoustic imaging system to image these agents. And we're doing this at 800 nanometers with laser fluence of about 5 millijoules per centimeter squared. And this is a safe laser limit. And so we've actually combined the magnetic targeting, so to speak, and the photoacoustics. So in this image here, we let the red blood cells settle to the bottom of the tube, and then we image them with the photoacoustics. And so you only see a signal down at the bottom of the tube. But then later we place the magnet on the top of the tube, and you can see that the magnet is bringing up the red blood cells to the top of the tube. And so this is just a very preliminary confirmation that this magnetic targeting, there might be something to it, and then we can actually visualize this effect using photoacoustics. And so in conclusion, we've made these constructs, the droplets plus the red blood cells. We have demonstrated red blood cell lysis by the vaporization of these acoustic droplets. And we've also demonstrated that photoacoustic imaging is a possible imaging modality for tracking these potentially in vivo. And we can do it with an FDA-approved drug. And so that's quite important. And then finally, the overall conclusion is that these may be a feasible alternative to some other drug carriers for image-guided drug delivery. And so with that, I'd just like to thank the members of the lab, also Sasha Klebanov, who's contributed greatly to this project, and then the sources of funding. So with that, I'll take your questions.